We're talking about the specimens and released over 150 preclinical models. I will try to summarize right now where these numbers are coming and what we can offer within CBTN portfolio. As I mentioned before, tumor models are derivatives of the tumor tissue. Obviously, we also bank primary cells within that effort. And just wanted to briefly present how here at CHOP we attempt to generate the models, which I'm going to be talking today about. So from the first issue, primarily maximize the effort to build up the organoid models currently, and that involves instant tissue microdissection and refers to what Jay and Dr. Santi were mentioning before about quick processing. As for the cell line development, the tissue undergoes the dissociation and mechanical processing in order to provide primary cells, which we also maximize banking. And then from that, we attempt to develop the cell lines and xenografts. Cell line-wise, we attempt 2 to 6 different combinations of serum-free media, depending on the number of cells obtained. And we also try to maximize use of the tissue using an explant-like approach for the serum growth. And sometimes we're very successful. This is an example of the case where we were able to develop all 3 types of models we're trying to share with the community. And organoid is really relatively a new approach which we started literally 2 years ago. And I'm going to talk a bit more later on about this process. But here I wanted to pause for a minute and indicate a general concept that our main goal is really to reduce the time from which our models or the models within CBTN can reach out to investigators. So quickly for that reason, whenever we observe the growth of cells in vitro culture and have several flasks of growing actively cells, we maximize the banking at that point, perform just our standard quality tests. And at that point, the cell line really enters so-called provisional model list. And officially, we don't share these on our list with investigators until specific models are of interest. And we're willing to provide those as provisional testing models where we hope that investigator can, if they're better specialists in specific histology, they can develop better model and return that to CBTN. For selected histologies, and we admit that we prioritize non-serum growth models, we attempt further thawing and propagation effort. So in our hands, we observe that even though the initial cell line is able to grow, after banking, it's not always easy to reestablish the growth. And obviously, it's not surprising from the experience of many of you. Once the cell line actually reaches logarithmic growth and we are able to propagate that, this is the banking event where we again perform our QC testing and the cell line ends up on the shareable model list. Depending on the, of course, capacity and time with start characterization, however, at that point, very transparently, we offer those models for the community. So looking at the numbers we delivered to the community so far, we had 50 cell line requests over time, and the interest is growing, which is really makes us very happy. Till now, in this year, we already obtained 13 requests. And just some statistics, not surprisingly, most of the cell lines which are requested from the community is really related to higher grade tumors. And the top of the list is high-grade glioma and ATRTs. However, all of, ependymomas, craniopharyngiomas, meningiomas as well are on this list. There is an interest, and we have more of those models within provisional list. We just don't have always capacity to propagate them, as their doubling time is extremely slow, and in many of them are actually growing better in the serum conditions, and they not always provide the best models. So between STGIDs, there is actually a one winner. The cell line 7316-3058 is actually the most shared aliquot and cell line across CBTN. I have actually personal story around this cell line, and maybe Angela remember that one night, or maybe not, but that was late in the evening, as Dr. Santi mentioned, as usually fresh tissue comes to the lab. And I think Marty and me were the only ones still on the floor. It took, before we shared this first time and banked the shareable model, it took us over 8 months to really call this cell line for the first banking. Nevertheless, it's not surprising that it's relatively often requested model. This is a screenshot from PCBio, where we actually have genomic data around that model together with the genomic data from the tissue. It's a progression case for high-grade glioma, and it has quite high mutation burden. Currently, we're working on the preclinical tumor models for CBTN. It's actually an app which is already active online, and we present that to every investigator who reaches out to us for the available models for the distribution. You can find here over 130 models for delivery. We're trying to annotate best to our knowledge what we know at for those models at that time. Looking at this one model, you can see that we have this one subject. You can see that we have 3 models available in 2 different medias. This is actually a modified model already with GFP and look construct as well available to share. But if you click at this hyperlink, this will basically direct you to the PCBio portal. So before you can actually see directly the genomic data about this cell line. We expect that currently, as I say, we directly offer this link to all of the investigators who are reaching out for the cell lines, but our goal is to make this Shiny App more comprehensive and listed on the CBTN cell line CBTN website as well. And another aspect which we really hope to achieve at some point is be able to list the cell lines not only coming from CHOP, but also donated or being able to be provided for other sites within CBTN. And currently we already started listing some of the models which we propagated for the medulloblastoma pedexis donated by Dr. Robert Westler-Rea. So this is the end where I come back for a moment for EXO-1, because that study actually has 80 cell lines genomic data coming and soon to be available within PCBio as well as raw data, obviously, as was mentioned before. And between those, again, we have majority of high-grade gliomas, some ATRTs, ependymomas, and a few other histologies as well. The beauty of that genomic collection is actually the fact that many of them at the same time and time point, materials from the same exact event were sent to PROCAN analysis as well, shared with Ron's group around childhood cancer model Atlas, and sent to NCATS for the drug testing. So shifting the gears for a moment for the organoid culture, I'm gonna just highlight what Dr. Storm and Dr. Santi were mentioning before. Whenever she says that she calls me for tissue, actually we taking it very seriously because she has really expectations that we're gonna be there within a few minutes, and we are. So for most of the tissues, we wanna make sure that within 30 minutes, the tissue ends up in hands for microdissection. And This project came out from our interaction and collaboration with Dr. Honjung Son from UPenn, who actually published very exciting work around GBM organoids. We utilized this one platform to all of the histologies, to tissue from all of the histologies which we received. And with lead of Valerie and quite a work from Rod, we're able to bank several organoids so far. And these organoids, interestingly, are able to being banked and then recultured again and grow for quite some time. The major difference between this model and cell line is that eventually organoids will stop growth. That's why they are extremely precious. This is a very unique case, which basically within 12 days obtained this very roundish bubble-like structures, which you can see by naked eye and under the microscope. There is no full diagnosis on that case. At that point, there is expectation of some high-grade tumor. And so what excites us the most is actually that we were able to try several low-grade glioma cultures on that platform. And this is exactly something what you might see on a poster tomorrow. Rod will be having more comprehensive information about those cultures. But as you can see, we have this one low-grade glioma case, which is KIA-BRAF positive, and the staining shown here at day 0 and day 40 indicate relatively similar composition of these organoids. Interestingly, we see immune component for several days in these organoids. But for this specific case, we are relatively sure that even after 40 days, there's some indication of microglia within this culture. So that's so far summarizes all the numbers which we processed. It's close to 40 cases. The failure rate is relatively low. Most of these at least were able to round, and we maximize the banking at early stages, looking really for the interest for also other investigators for utilization for those. How we actually expect that this might have more translational meaning is really by the fact that the tissue really ends up in our hands quite quickly. The process of genomic analysis on the clinical side takes several weeks. So knowing that our culture can grow and form really growing model within 2 to 3 weeks in many cases, this might be enough to generate the molecular data, provide the tumor profile report, and then identify targets which we could explore for therapeutic testing within the lab space. And together with a genomic report and organoid drug testing report, we hope that that would end up at molecular tumor board and inform the clinical decision. And again, this takes an army really on the neurosurgeon's effort, and Jay's group, Peter Madsen, Alex Tucker, Shin Ben within CHOP, pathology effort. But I wanted to here specifically highlight all of the folks who really put a lot of work at the late evening in the lab. As you probably heard that Dr. Santi often calls us at 7, but it's not trivial. We often put this work at that late time and make sure that we can maximize development of those models. Thank you.